This is part 2 of the chapter 13 from Campbell's Biology for AP Biology. This is about crossing over as it refers to meiosis and sexual life cycles. So remember we talked about meiosis and the process of undergoing meiosis to reduce the chromosome number. The things that you need to keep straight about meiosis are the events that are unique to meiosis. All three of these events occur in meiosis 1. We have synapsis and crossing over, that's producing the tetrad and of the homologous chromosomes, and it lets them physically connect with each other and exchange genetic information. The second event is when the paired homologous chromosomes or tetrads line up at the metaphase plate. They're all there together rather than being individual chromosomes. They're replicated. And then in anaphase 1, the homologous chromosomes separate rather than the chromatids. This is definitely important to remember about. All of these things happen in meiosis 1. The thing that allows the chromatids to stay together is something called sister chromatid cohesion. There are proteins called cohesins that hold the chromosomes together. Now in mitosis, those proteins are broken down at the end of metaphase. But in meiosis, they only are broken down on the chromosome arms in anaphase 1, but not in the centromeres. They don't separate or break down from the centromeres until anaphase 2, when the chromatids separate. In the process of this sexual life cycle, of course, the variation contributes to evolutionary process because there are mutations, of course, that are the source of all genetic diversity. Basically, they create different versions of genes. The genes are called alleles, and then reshuffling those alleles allows for genetic variation within the species, which is very important overall to the survival of the species in terms of having lots of different combinations of genes that allow for survival under different conditions. In meiosis and fertilization, of course, this is what happens with the chromosomes is what allows for this variation in each generation. There are 3 mechanisms, and we'll talk more about these mechanisms in genetics, but they contribute to the genetic variation that exists. First of all, the independent assortment of chromosomes, crossing over, of course, and then random fertilization. And so let's talk about each one of these in turn. Independent assortment is one of the laws that Mendel proposed about how the genes did not, the inheritance of one gene does not influence the inheritance of another. And we can relate independent assortment to what happens with the chromosomes here in meiosis. Those homologous chromosomes orient themselves randomly at metaphase 1 of meiosis. And so in this independent assortment, what it means is that each pair of chromosomes sorts out those maternal and paternal chromosomes into daughter cells independently of all the others. The inheritance of one is not influenced by the inheritance of a different one. They're just randomly sorted there. There are a number of different combinations present, and that depends on how many you can have, depends on how many chromosomes there are. The mathematical formula is 2 to the nth power, where n is the haploid number of chromosomes. So for humans, since n is 23, then there are more than 8 million possible combinations of chromosomes. 2 to the 23rd power is something like 8,400,000. And so there's a lot of variation there in the ways that those chromosomes can be assorted independently from each other. Kind of boggles the mind to think about it. So here's a simplified version to show you if you had 2 pairs of chromosomes. And so we get 2 possibilities of the way they could line up at metaphase, either with both paternal chromosomes on the left and both maternal chromosomes on the right, or they could reverse themselves like that. All right, and so in metaphase you could separate those homologs from each other, and you end up with 4 possible combinations here. Both chromosomes from dad, both chromosomes from mom, the long one from dad, the short one from mom, or the long one from mom and the short one with dad. So we have 4 different combinations that you can have there. And that you can see how as you increase the number of chromosomes, you have more and more combinations, which leads to a lot of possibilities for variation. Crossing over is the second thing that influences the variation that exists in the offspring. And that's because you get those chromosomes get recombined with each other, and that combines DNA inherited from each parent. It starts very early in prophase 1, and those homologous chromosomes, you remember, pair up gene by gene in prophase 1, very early in prophase 1. And as they do this, they can start crossing over. We have homologous parts of two non-sister chromatids that trade places with each other. Basically, part of the DNA breaks off of each and reattaches to the other. And this increases genetic variation because it combines the DNA from two parents into a single chromosome. So here you have your non-sister chromatids that are held together during synapsis. You have the crossing over that occurs here. This particular picture shows you two chiasmata there, but they can have many more than that depending on lots of different factors. I mean, considering how long some of the chromosomes are, you can see how they could cross over multiple times. And so you could end up with a great deal of mixing between the chromosomes from one parent and the other. This particular one just shows a very simplified one where they cross over twice. And so you end up with 4 unique chromosomes that are passed on to the offspring. 2 of them here it shows would be non-recombined chromosomes and 2 would be combined, but that doesn't mean that every chromosome any chromosome in particular is going to be passed over uncombined, because they can recombine several times. The third thing that contributes to the great deal of variation is something called random fertilization, and this is going to add to variation because any particular sperm of the millions can fuse with any ovum, and when those gametes combine, and remember we said that there are 8.4 million, 800,400,000 possible chromosome combinations in humans from independent assortment, then when we put those two gametes together, we get a zygote that can have any of about 70 trillion diploid combinations. It's just, you can't even imagine numbers that high, basically, when you think about the combinations. So it's amazing that we even have any resemblance to each other at all, because there's so many possible combinations here. That's not even counting crossing over. So when you add crossing over to it, of course, you've got a lot more variation that can be there. So that gives each zygote a unique genetic identity. Now you might be thinking, wait a minute, how do identical twins form? Well, of course, identical twins form from one zygote that during the process of embryonic development splits into 2 separate groups of cells that each develop from the same genetic source. And so that's why they can be identical, only because they start from one zygote. This is really important in terms of the genetic variation within a population, which is a very specific and a very, very significant thing and very necessary to natural selection, of course. So what we're going to have is the accumulation of these variations that are favored by the environment. Sexual reproduction is going to contribute to that variation, which is going to ultimately originate from mutations, and that leads to lots of different kinds of combinations that can be present in the population. And we'll talk a lot more about the significance of this and how this influences the inheritance of traits and influences the evolution of a species entirely.